Now, this is how the cerebral cortex, the dissection of the cerebral cortex looks like. Look up here. This is the, what is it? This is the outer part of the cerebellum, right? This is the cortical part of the cerebellum. What is there deep down? This is the white matter. Okay? The white matter of the cerebellum. Remember, deep in the white matter are present the deep nucleus of the cerebellum. The deep nuclei, they will come back again. Okay. Now I am going to do, what have we done here? We have taken a section of this and magnified it. Once we magnified, what are we getting? We are getting these layers. Okay. Before you actually get confused with this, let me take a blank slide. Let's go to the layers of cerebellum. So for layers of cerebellum, there is a mnemonic. You should remember. What is that? My pretty garden. My means molecular. P stands for Kenzie. Don't say it's a pretty. And G, the garden, G stands for granular. So we basically have 3 layers, right? So we are going to make that one. This is my molecular layer. Good. This is my Purkinje layer. Okay. And this is my granular layer. So this is my molecular layer. This is my Purkinje layer. This is the granular layer. Now let me take some important structures first. So we'll try to understand first the basic one, and then we'll go and complicate it more. Remember, if, this is the cerebellum, this part is the cerebellum, all right? Cerebellum gets multiple inputs from all over the places. It gets, as we know, it is getting the input from the cortex. It is getting the input from your vestibular nuclei. It also gets an input from the spinal cord. It gives an input to the spinal cord. So a lot of connections are there, afferent and efferent fibers. Let us go with the fibers which are coming to the cerebellum. Remember, except for one nuclei, what do we call that? That's called as olivary nucleus. This connection which comes from the olive to the cerebellum is called as olivocerebellar pathway. Except for these fibers, rest all the connections, all the fibers, they have got one name. They have a single name, and that name is the mossy fibers. So what are my mossy fibers? What about this fiber? This is called as climbing fiber, right? Let me tell you what is the difference between climbing fiber. Climbing fiber is denoting to the connections which is there from between the olive and the cerebellum. That connection which is coming from the olive to the cerebellum is called as a climbing fiber. Rest all other who are entering into the cerebellum, they all are divided as mossy fibers. They're classified as mossy fibers. Okay. So with that in mind, let us start with the tracing. Remember, all other fibers, whether it is coming from the cerebral cortex, whether it is coming from the vestibular apparatus, whether it is coming from brainstem, whether it is coming from your spinal cord, they all will be called as mossy fibers. And how about the climbing fiber? Climbing fiber is specifically denoting the connection from the olive to the cerebellum. Okay, except for that one specified special pathway being named as climbing fiber, rest all are called as mossy fibers. So let us trace these pathways and make the circuit. Okay, so let me take a deep nuclei. From the deep nuclei, we'll have the outputs. Okay, be very happy. This is my major output. And what about my inputs? I had 2 inputs. What were those? One was the climbing fiber. The best part is, as these climbing fibers are entering into different layers of the cerebral cortex, they give collaterals, or you can say they synapse with the deep nucleus, and this synapse is excitatory in nature, and the neurotransmitter which is released here is aspartate, right? How about mossy fibers? Okay, So let me trace a mosey fiber also. Okay, let me take another color here, maybe the green one. So mosey fibers, this is my mosey fiber. And this is my climbing fiber. Don't forget, climbing fiber is coming from the olive. Mosey fibers are any other fibers, right? So mosey also, when it is entering into the layers of cerebellum, it gives a stimulatory connection to the deep nucleus of the cerebellum. This is called as the deep nucleus, right? And the neurotransmitter which is attached here, which is released here, is your glutamate. Okay. Now, look at the fun. Let us now draw the other cell components. It is a Purkinje layer. So definitely what do we have? We have the Purkinje cells. So this is the structure of the Purkinje cells. This all are the dendrites of the Purkinje cells. These are the dendrites of the Purkinje cells. So I'm just drawing to make you understand 2-3 of them. Good enough. Now let us take these Purkinje cells. The best part is this is the dendrite of the Purkinje cell. The axon of the Purkinje cell, it comes and synapses with my deep nucleus. And this is inhibitory in nature. If it is inhibitory, what is it releasing? GABA. Okay. Now, this is the cell layer. Definitely in the granular layer, we will have cells later on. We'll see the granule cells are there and all that. Look at the connections first. What is the connection here? The climbing fibers. What is their role? They go all, I can take the other color. Okay. So these climbing fibers, when they go up where? They ascend up, and they go and stimulate again whom? The dendrites of the Purkinje cells. Okay. Now, this pathway, I'll come back again and let me show you here. Get familiar here. So what are the layers? If I look at the layers, we have he has gone from my molecular layer, the outermost layer, then a thin Purkinje layer, cell layer, then comes the granule cell layer. Okay. The fiber which is coming from the inferior olive, that is very special because they have got a special nameplate. This is called as the climbing fiber. These climbing fibers, they are going towards the Purkinje cell layer, or you can say towards the molecular cell layer. What do they give? They give collateral. They stimulate the deep cerebellar nuclei. Things to remember here is, what is the neurotransmitter which is released? For your information, the neurotransmitter here is aspartate, right? And what is happening? These climbing fibers are stimulating whom? Look at this. They are stimulating, the climbing fibers are stimulating my dendrites of my Purkinje cells. How about the other fibers? The other fibers whom we call as the mosey fibers. Mosey fibers may be any fibers coming from pons, vestibular apparatus, spinal cord, etc. What do? They also go and give a stimulatory fiber to the deep cerebellar nucleus. And the neurotransmitter which is released here is glutamate. Look at the Purkinje cell. The Purkinje cell, it also, the axon of the Purkinje cell, it comes down and gives a synapse to the deep cerebellar nucleus. And this synapse is actually inhibitory. So the neurotransmitter which is being released here is the GABA. Okay. What is released here is GABA. And what is the neurotransmitter here is glutamate. Right. Now, let me tell you here and then we can draw it there. What do we see? The mosey fibers, they go and give multiple synapses to the granule cells. Okay. We have the granule cells and the granule cells are going to give the parallel fibers. Let me draw it in our diagram. So, let us take the granule cells. So, let us imagine this is a granular layer. We have the granule cells. So, we have many granule cells, right? How about, and they are giving lot of dendrites. There are lot of dendrites here. What is happening with my mossi fibers? They are going and making a connection with these granule dendrites. So multiple connections are made. What is difference between the mossi fiber and the climbing fiber? The difference is climbing fiber has one-to-one communication. One climbing fiber is going to communicate with single Purkinje cell. But one mossi fiber may go and communicate with many granular cells. Okay. Here also what is the neurotransmitter which is released here? The neurotransmitter released here at this synapse is also glutamate. Okay. Now, the funny part. What is that? The thing is that these granular cells, they ascend up as parallel fibers. They all ascend up as parallel fibers. And as they reach towards the outer part, they bifurcate and go along this is how they go, parallelly. Now, these are called as parallel fibers. These parallel fibers, what do? They go and they also make connections with the dendrites of the Purkinje cells. So, they are making connections with the fibers here. They make connections with the fibers here. Right? So, this part, the parallel part connection, let us understand from here now. So, what do you see? The mossi fibers, it goes and one mossi fiber, see, one mossi fiber can go and innervate many granular cells. And these granular cells are going to give what? Parallel fibers. These are your parallel fibers who are going to go parallelly along the outer part of the cortex. They are going to go parallelly like this. And they are going to make connections with whom? With either the Purkinje cells. So they may make connections with the Purkinje cells. Look at this. There is one more thing that we have to know. There are other cells, what we call as the Golgi cells. Okay, they may come. Then we have the basket cells. Then we have the stellate cells. That we are going to study for now. What is important to know is these parallel fibers, they go and make synapses, or you can say they stimulate the Purkinje cells. If they stimulate the Purkinje cell, now look at this. Cerebellar fibers, the climbing fibers, the input fibers which are coming from the olive on their way to the molecular layer, they stimulate your deep cerebellar nucleus. The fun is they also stimulate the Purkinje cell, which in turn inhibits this one, deep cerebellar nucleus. That means directly I am stimulating, but indirectly I am inhibiting. Why all this is in the central nervous system is this indirect inhibition is basically going to fine tune the outputs from this one deep cerebellar nuclei. How about here? These parallel fibers, they are going to stimulate again these parallel fibers. So this was stimulatory. This was stimulatory. Glutamate was being released here. Now, as these parallel fibers, they ascend up, they go and give collaterals and they are going to synapse, make connections with the Purkinje cells. These connections are again stimulatory. That means they are stimulating the inhibitory Purkinje fiber, which in turn again is going to inhibit my deep cerebellar nuclei. There is two more nucleus. Now the fun is that we have two more nucleus. What are those two nucleus? One is called as basket nucleus. One is called as the basket nucleus, and there is one star-like nucleus, and we call that nucleus as stellate nucleus. Okay. Now, what is the fun is that these are inhibitory to Purkinje. Basket cells are inhibitory to Purkinje. Stellate is inhibitory to Purkinje, and they are stimulated by my parallel cells. Look at the fun of it. Why all these is happening? This is happening because Ultimately, your brain has to get this output. Okay. And these all are inputs which are coming to the cerebellum. Now, whatever is the input, the whole thing is that it has to be very finely and very meticulously, has to be coordinated, has to be fine tuned. Now, if I want that if there are, let me take like this, if these are there, these are many Purkinje cells, okay, they are making inhibitory connections with the deep nucleus. These are the dendrites for these Purkinje cells. There is another Purkinje cell. These are dendrites for that Purkinje cell. That is also coming. Now, suppose I want that my impulse from the central Purkinje cell should reach, okay, and the rest of the information coming from the peripheral part should not reach to my deep nucleus. What should I do? I should inhibit the neighboring one, right? Then only the information which is passing from the central one, from the major one, will be executed. It's like I want to be projected. Suppose I'm in a GD, group discussion, and I want to project myself only in that small group discussion. What should I do? For projecting myself, I have to suppress my neighbors. If I do not suppress my neighbors, I will not be projecting myself, not showcasing myself. Similarly, in central nervous system, if you want that I want this input to be sent, the signal, I want to identify the signals which are coming from this central part, there has to be inhibition of the neighboring parts. How is that possible? That is possible by inhibiting them. And who is going to inhibit the neighboring ones? Inhibition will be via the basket cells. Inhibition will be via the stellate cells. So basket cells, stellate cells, they are going to inhibit the neighboring Purkinje cells, and they may leave the connections, they may leave the inhibition from the central one, so that I am able to identify these signals from this are only sent as an output. Ultimately, they are going to modulate the deep nuclei impulses, and deep nuclei in turn is going to modulate my efferent impulses. All right. So this is the connection, simple connection of cerebellum. But there is something more interesting which we have to see in this slide. So again, we are revising. It is worth it to revise because this comes as a question. You can say cerebellar circuits comes as a question, right? Now see here, again, we have the deep nucleus which is present there, and we have the fibers. Who's the input fibers? One input fiber is from the climbing fiber.
Climbing fibers.
And the other input via the mossy fibers.
Mossy fiber.
Yes, the climbing fiber is my major fiber, means coming from the olives, have got a special name, and there is a connections for that. Then it goes and stimulates the Purkinje cell. Purkinje cell in turn, the axon is going to be inhibitory to the deep nucleus. The part which I wanted to show you here is this. This is called as glomerulus. Glomerulus means it forms a bulb-like structure. Why? Because here is where we have the multiple connections between the granule cells, the mossy fibers, and the granule cells. It is kind of, see here, the granule cells, dendrites. How are they kind of interacting with the mossy fibers? As climbing fiber is one-to-one connection, mossy fiber is one-to-many connection.
One-to-many.
Okay, so look at it, how one-to-many. This is my mossy fiber. Okay, when this mossy fiber comes, we have many granule cell dendrites who are coming. Not only granule cells, we have another cell which is called as the Golgi cells. They are also inhibitory. Now these Golgi cells also come and synapse with my mossy fibers. Granule cells, mossy fibers is going to stimulate the granule cells. It is also going to stimulate the Golgi cell axons. Golgi cell axons probably will remain in the granule and the Purkinje cell layers. But what is important is my granule cells. Granule cells are going to ascend up as parallel fibers. Look at this. This is how we have, as if somebody has made a shed, parallelly they have arranged the bamboo sticks like this. There is going to be parallel fibers. And what is the role of these parallel fibers? they are again going to go and synapse with the dendrites of the Purkinje cells. They are going to go and synapse with the basket cells. They are going to go and synapse with the stellate cells. They actually are going to stimulate the stellates also. They will stimulate the basket also. They will stimulate the dendrites of the Purkinje cell. But the problem is, once you stimulate these cells, Purkinje is inhibitory to the deep cell nucleus, basket is inhibitory to the Purkinje, stellate inhibitory to the Purkinje. Okay, this is how the whole circuit goes. Look at this. Again, I'm trying to explain it. This is the input. Okay, input to the deep cell nucleus, deep nuclear cells. Then we have the inputs to the Purkinje cells. Purkinje cells obviously are getting stimulated, but in turn, Purkinje cell is inhibiting my deep nuclear cell. That's an inhibitory input from the Purkinje cells. How about Mosey fibers? Mosey fibers. Ankit, can you mute? We have sounds coming from your side. The Mosey fibers, they are stimulatory to the deep nuclear cell, as well as they are going to go to many granule cells. Many granule cells are going to form the parallel cells, and these parallel layers again are going to stimulate my Purkinje cell. Purkinje cell in turn is going to inhibit my deep cell nucleus, deep nuclear cell. Okay, so deep nuclear cell has got excitatory afferents coming from the climbing and from the Mosey fibers, and an inhibitory input from the Purkinje cell. Now, what is it going to do? It is going to, this excitatory and inhibitory coming to it, ultimately then anastomosing here. They ultimately are going to have what? This deep nuclei is going to calculate both excitatory inhibitory effect, and a fine impulse, a fine tuned output is going to go out of the cerebrum.